There are thousands of different enzymes inside your cells because each enzyme is a specialist in performing one and only one particular function, particular chemical reaction. But taken all together, all those chemical reactions that all your enzymes carry out are necessary to keep your cells alive and functioning. We also have receptor proteins. They are embedded in the membrane, and part of them sticks out of the membrane, and their job is to detect what molecules are floating around outside the cell. There are also transport proteins, which are sometimes called channel proteins, and they are responsible for moving solute molecules in and out of the cell. And just like the enzymes, each receptor protein and transport protein is a specialist in just working with one and only one particular molecule. Okay, now here I've been showing the proteins as little worker dudes, but of course they are proteins, which means that they are chains of amino acids. And so this would be a more realistic view of what the proteins look like. And remember, this part right here is called well, in enzymes it's called the what? The gap where it binds its molecule is the what? Anybody recall? It's right. In enzymes, the area where it binds the molecule it works with is called the active site. But in receptors and transport proteins, it's called the binding site. Yeah, two different terms, but it essentially means the same thing. Good, and of course the little balls are the amino acids, which are the monomers that proteins are built out of. And something I mentioned a while back is that the sequence of amino acids in a protein is what makes it a specialist. The reason why one protein differs from another is that they have different sequences of amino acids. And the reasoning behind that is this: the sequence of amino acids is what controls the folding of the protein, and since the folding of the protein is what forms the active site or the binding site, that's why the sequence of amino acids essentially dictates what the protein's function is. It controls the folding, which makes the binding site. Okay, what I want to talk about now is how the cell puts together proteins. To make a protein, the cell has to link together amino acids. And there's an organelle inside all cells, whether it's a prokaryotic cell or a eukaryotic cell. They all have this organelle, and that organelle is called a ribosome. The ribosome is the organelle that links together amino acids. Here's a close-up. Now, here in this cartoon, it looks like the ribosome is almost as big as the nucleus, and that's absurd. Ribosomes are a lot smaller than the nucleus is. You couldn't see individual ribosomes under one of our microscopes here. You could see the nucleus pretty easily, but the ribosomes are just way too small. But just so you can see that it's made it a big ribosome. And I'm only going to show one ribosome here, but cells have, I don't know, hundreds if not thousands of ribosomes floating around inside them. So a ribosome is an organelle, and it's made out of partially protein and partially RNA. Parts of the ribosome are protein and parts are RNA. Ribosomes have two subunits. One part's called the small subunit here, and one part's called the large subunit here. And there's a little gap between the two subunits of the ribosome. And so as I said, the job of the ribosome is to make proteins by linking together amino acids. Where are the amino acids? Well, the amino acids just float around in the cytoplasm. In other words, in the watery liquid that fills the cell. There are lots of dissolved molecules in there, and some of those molecules are individual amino acids. So when the ribosome wants to make a protein, it just essentially grabs amino acids that are floating around and links them together, makes covalent bonds between them, and that's how a protein is made. If you notice, the protein is being constructed in the gap between the small unit and the large subunit of the ribosome. Speed up that process a little bit. Go faster, go faster, go faster. There we go. Almost looks like a little caterpillar or something. Now, here's the ribosome. And poor Mr. Ribosome looks a little frustrated and confused here. Oh yeah, these are the each amino acid has its own particular name. There are 20 different amino acids, and each one has its own particular name, which is what these are. Anyway, why does the ribosome look frustrated? Well, because ribosomes are workers. Their job is to link together amino acids to make proteins. But a construction worker needs a blueprint to follow. You can't just hire a construction worker and say, build. The person would say, well, what do you want me to build? I need some sort of plan. I need a blueprint. And so, yeah, ribosomes put together proteins, but the ribosome needs to be told which particular amino acids it has to link together in which particular sequence to make any particular protein. And that's where the DNA comes into this picture. The DNA over here inside, which is part of the chromosomes, which are inside the nucleus, the DNA contains the genetic information to operate the cell because the DNA contains or is the instructions for making all the cell's proteins. So when I've been saying the DNA is the blueprints or the genetic information, what I mean is this, that the DNA is the recipe book that holds the recipes for making all the cell's thousands of proteins. And that's how the DNA controls the cell. It's the recipe book for the proteins, and then once the proteins are made from the information in the DNA, then those proteins actually carry out the tasks that run the cell. But the control center, the information is stored, the recipes are stored in the DNA. And so what I want to do is look at that process, the interaction between the DNA and the nucleus and the ribosomes actually making the protein, and see how it works. And to illustrate this, we're going to have the ribosome make a relatively small protein. This protein is going to start off with the amino acid methionine, followed by the amino acid glutamate, and followed by the amino acid proline. That's a very short protein, only 3 amino acids long. Most proteins are on the order of 400 amino acids in length. But just for illustration, let's just pretend it's a very, very short protein. Okay, so the ribosome pieces together the amino acids to make the protein, but the recipe for that particular protein is stored in the DNA in the nucleus. And so let's move over to the nucleus and take a closer look there. All right, so there are the chromosomes inside the nucleus. The segment of DNA on one chromosome that encodes a protein, that holds the recipe for the protein, is called a gene. And so we'll say, for the sake of argument, that this segment of this particular chromosome is the one that makes that protein we just saw, or that I should say that encodes that protein that we just saw. So this is one of the genes on this particular chromosome. But chromosomes are very long pieces of DNA, and they hold more than just one gene. For instance, over here on that same chromosome is another gene that holds the recipe for a different protein. Over here is yet another gene on that same chromosome that holds a recipe for another protein. Another gene is there, and you have more than one chromosome, and each chromosome typically holds hundreds of genes. Now, human beings have around, well, we have exactly 46 chromosomes in our cells. And we have around 25,000 genes total. And, I've never done this calculation before, but how many genes per chromosome on average? I guess 25,000 divided by 46. Looks like an average of 543 genes per chromosome. And that's just an average, because some chromosomes are very small and probably only hold 100 or fewer genes, and some chromosomes are large and probably hold over 1,000 genes. But it looks like on the average about 500 genes per chromosome. Okay, now let's just zoom in on that one gene that's going to encode that particular short protein that we just talked about. And let's say it's right here, there. All right, well, let's look at this gene a little more closely, where we can see the actual G's and A's and T's and C's on it. So well, you know DNA is double-stranded, double helix, and if you have a G on this strand, it'll always be complementary base pairing with a C there or a G there, and A's and T's are always opposite each other on the two strands. Just to make the diagram a little more understandable, I'm going to kind of straighten out the double helix. In real life, of course, it would still be twisted around in a helix, but just so we can see it better, let's look at it like this. All right, so here's this gene that we're talking about. There we go. Here's how genes encode the amino acid sequence of their protein. Each 3 consecutive nucleotides in the gene, in other words, each 3 neighboring nucleotides, is called a codon, and each codon stands for one of the 20 amino acids. And to make a non-biological analogy here, if we have the letters CAT, to us that represents something. That represents that. And if I take the 3 letters DOG, to us that represents something. It represents, well, it's supposed to be a dog. And so what I'm saying is, to the cell, each 3 letters, each codon of the DNA is like a little word that stands for, that represents one of the 20 amino acids. So the sequence of codons of 3 consecutive letters in the gene spells out to the cell a recipe for a particular protein. Where did I put it? Here it is. Well, how many codons can there be? Well, the answer to that is 64. If you think about it mathematically, in any particular spot, there could be any one of 4 nucleotides, A, G, C, or T. So there's 4 choices there. The second nucleotide of the codon